Okay, so we had innate immunity and now we have adaptive immunity. Adaptive immunity is going to involve some specificity, okay? Specific lymphocytes are going to respond to a specific antigen, all right? That's how it works. We have to come in contact with the foreign invader, we proliferate in number, and then we mount the immune response. And we have a number of antibodies that are going to help with our immune response, again, responding to a specific foreign invader. The thing to remember when it comes to adaptive immunity, it is going to take a little bit longer. That's probably the one thing that does separate it from innate. With innate, it's just non-specific. We have all of our various different general type of cellular activity that just goes in and engulfs. To engulf something is really not very specific ultimately. If it just sees, oh, hey, foreign invader, chomp, not very specific, all right? So for adaptive immunity, it is going to take a little bit longer. It can take days to develop. It can take a few days for the system to potentially even see the specific foreign invader, and then several days after that to mount the immune response, and then it could take upwards of 2 weeks to maybe get rid of it. So it does take some time when you are just first getting exposed to this foreign invader. As you can see, it takes days to develop, and it is considered the third line of defense, and we just talked about the first two. When we look at our different types of adaptive immunity, we have cell-mediated immunity and humoral immunity, and this is antibodies, okay? B lymphocytes is humoral immunity, okay? Make plasma cells, and plasma cells give rise to antibodies. And we have 5 different types, and we'll look at those at the very end here. Cell-mediated immunity involves T lymphocytes, okay? So what these ultimately involve are very different. Cell-mediated immunity involves the T cells, the T lymphocytes. Humoral immunity is uber specific, and it is going to be the B lymphocytes specifically making antibodies. Okay. And here's cell-mediated immunity, T lymphocytes. Okay. And then here's our B cells, our B lymphocytes making plasma cells, plasmacytes, which then make antibodies. Okay. There are, as you can kind of see here, we have T cells, we have cytotoxic T cells, we have helper T cells, we have T cell regulators. We have a lot of different types of T cells. As you can kind of see, as you can imagine here, cytotoxic T cells kill, helper cells, T helper cells help the cytotoxic T cells. And they do that by, basically, calling up their buddies, the B cells, and being like, hey, I got this thing I can't get rid of. I need your help here. So T helper cells can call in the B cells, which goes from cell-mediated immunity to antibody-mediated immunity. And the body can easily switch back and forth between those two. What are we attacking? We are attacking foreign invaders. What do foreign invaders have? Antigens. What do all cells have on their surface? Antigens. Antigens are the markers on the surface of cells that allow us to see us or see the foreign invader, okay? And this is what really everything is going to bind to, whether it be a T lymphocyte or an antibody, which we'll talk about briefly with the antibodies functions here in a little bit. But, it's a large protein, and it's the protein markers on the surface of it. And so whether you're a T lymphocyte or you're an antibody, you see it, and you're basically going to attach to that thing. Some examples of antigens are viral or bacterial or fungal or parasitic in nature. And, there's a number of things that can be a foreign marker that the body will mount an immune response against. The outer protein capsid of viruses. The capsid is what helps kind of protect the virus, allow it to survive on a surface for a longer period of time and whatnot. Cell wall of the bacteria. Some of your bacterial infections will have a cell wall around it. This just gives some further protection. Think of it like a cell wall in a plant, gives it some rigidity and some structure. Basically does the same thing here. The secretions of them, bacterial toxins, the foreign invaders, bacterial, viral, they usually produce a number of various different things that they secrete to further their agenda, and that's them invade, make cells more susceptible for invasion and whatnot. So number of different things. Bacterial toxins are just one of them. Abnormal proteins, there can be tumor antigens even that kind of inhibit some T cell activity. So pretty interesting what these foreign antigens can be. And here I kind of keep, recognizing, foreign and self. Foreign, again, these are just body markers that aren't ours, right? We have self antigens, they're our own body markers, okay? And typically our cells don't bind to them. They can. There's a number of autoimmune conditions out there, right? So that these aren't always fail-safe. There is some issues that can rise up, and we're not going to get into that because it's pretty complex. But foreign antigens, self-antigens, they're the markers that allow us to know us or you, the foreign invader. There is, as you can kind of see here, is a beautiful picture of our foreign invader, and there's all the little kind of antigens that are extending off of the plasma membrane of said foreign invader. All of these proteins that are projecting up off of the surface there are referred to as an epitope, okay? And these antigenic determinant sites are what give us the specific site that can be recognized by the immune system, so we can mount a specific immune response. Because the much more specific the immune response, the more effective the immune response can be, and the more targeted it can be, where we just are killing off the foreign invader and not necessarily all of the cells in the region. So it does give us some specificity in that regard. Each has a very different shape, and it is that different shape that gives it its ability to be so effective at the next season. It could be something similar, but we just change one little marker on that surface of that epitope, and it's like we've never seen it before at all. But all it is the flu from last year, but now instead of being marked on this side, it's marked on this side. And for that reason alone, our body is like, I've never seen this before, so now I have to go through the entire process of attaching to it, recognizing it, making memory cells, all that whatnot, so I can remember if that specific type of influenza comes back again, I can mount it a hell of a lot quicker and fight it off. So it is very specific, this epitope mounting a specific immune response. Immunogenicity is the ability to trigger the response. And if it's newly seen, how big is the antigen foreign invader? If it, one of the functions of an antibody is to cause agglutination or clumping. And with that, we increase the size basically of the pool of foreign invaders. So it becomes a much more effective immune response, immunogenicity, by just increasing the size of this thing. So the antibodies do that as a function to create a much bigger pool of foreign invaders there, instead of just one little foreign invader in 5.25 liters of blood. Okay, makes it a little bit harder to find that needle in the haystack. When the antibodies clump to it there, when attach to it, neutralize it, it can cause this thing to, clump and precipitate out of solution. And so we can mount a much more bigger immune response, have much more immunogenicity. Again, we have our T cells and our B cells. We have our T cells mounting cell-mediated immunity, and we have our B cells mounting humoral immunity or antibody-driven immunity. When we look at these T cells and B cells, T lymphocytes and B lymphocytes, they have very unique receptors on their surface, very complex. There, as you can kind of see, I know I don't usually ask the number of them. I just put it there. There are 100,000 of these receptor complexes per cell. So you can see that these cells, these T lymphocytes and B lymphocytes, are very complex. Very complex. They have a number of receptors on them, number of complexes on them, so where that they can attach to the foreign invader and really get this process of the immune response going as fast as it can. We have TCR and BCR. TCR is just T cell receptor, okay? And this is a TCR antigen receptor for the T lymphocyte, okay? If we have this receptor complex on the surface of this lymphocyte, it's going to be a T lymphocyte, okay? We have BCR, B cell receptor, and this is the antigen receptor that we find on B lymphocytes. And they are a little bit different, but that's really all I want to say about it. These receptor complexes are numerous, very complex, and depending upon the lymphocyte, what receptor complex the lymphocyte has on it, will dictate whether or not it's going to become a T cell or a B cell. All right, so lymphocyte in the blood comes in contact with the foreign invader, okay? Comes in contact with an antigen. B lymphocytes will make direct contact with the antigen, all right? We're gonna look at what ultimately antibodies do, and they have a very specific region that they will go out and touch and hold on to and capture the foreign invader. T lymphocytes usually have an antigen presented by another cell. So usually with T lymphocytes, it's like a macrophage or a monocyte that comes in contact with this foreign invader and says, hey, look what I found. Can you kill it? That's how T lymphocytes work. So they don't necessarily go out and attach to the foreign invader directly. They usually do it through another means. Usually someone else brings and says, look what I found, okay? We have several different types of T lymphocytes, and the two kind of most important ones are right here, okay? Helper T lymphocytes and cytotoxic T lymphocytes. Helper T cells, cytotoxic T cells, okay? Helper T lymphocytes are also referred to as CD4 plus cells, okay? Cytotoxic T cells are usually called CD8 plus cells, okay? I would know those if I was you. They usually get asked on exams, board exams, and stuff like that, from what I hear as well. These helper T cells, they assist in cell-mediated immunity. Okay, we just saw that picture. They help with humoral immunity, antibodies. And they help with innate immunity. So they help with all aspects of immunity. Cytotoxic T cells, these are our CD8 cells again, and these are the dudes or dudettes that release the chemicals that penetrate either the infected cell or the foreign invader and destroy it. Digestive enzymes that just cause destruction. And you can see CD4 and CD8, they do have very specific types of receptors. And there's the B cell receptor, okay, which is the antibody. And we'll talk about that variable region, a constant region, and what kind of goes on there. So when T cells get brought something, here it is. So how do these T cells present? Or how do they get presented with all of these foreign invaders? Well, it's called antigen presentation. Here's your sign, right? Duh. But when it comes to these things, sorry. We have basically two categories, and all nucleated cells of the body have antigens on it, and even red blood cells, right? But when we're talking about immune cells, we are talking about antigen presenting cells. What are those antigen presenting cells? Well, one of them are B lymphocytes. Okay, B lymphocytes are going to be able to differentiate into plasma cells and ultimately into antibodies, and they attach. And they will present it to other antibodies, mounting immune response. We have macrophages, and these are as basic of an APC as we got. Macrophages will engulf, and if they can't kill it, they present it to our T cells, to our T lymphocytes. Lastly, we have dendritic cells. And dendritic cells, we find those within the skin. So we have there right after that first barrier, that first boundary, that first line of defense, the skin. Okay, we have dendritic cells. As we get a little deeper, we got macrophages. And then ultimately our third line of defense, antibodies. So these are our antigen presenting cells: dendritic cells, macrophages, and our B lymphocytes. We have to attach, okay? We have to attach to the protein on that surface. So it requires what's called MHC, major histocompatibility complex. This is what we're trying to get as close to 100% match on when we do transplants, MHCs. These are basically what's going to help us to determine that it's us and not a foreign invader. It's just a group of proteins, as you might imagine. That's really all we're going to get into it. There are multiple different types of them. MHC1 is found on all nucleated cells, and MHC2 is found on specifically our antigen presenting cells. And that's really what I want you to know. That's what I would ask about this on the exam. What is an antigen presenting cell? Dendritic cell, macrophage, B lymphocyte. And what type of MHC do they contain? What type of major histocompatibility complex do they contain? MHC1 is found on all nucleated cells. MHC type 2 is found on all of our antigen presenting cells. So that is how it's going to attach and present to the T cells. When we look at lymphocytes, when we look at how they proliferate, it's a very, very complex kind of thing. And we're just going to keep it super streamlined, super basic. If you want to read more about it, the book goes into it in very good detail. All right. But what I have in the notes, that's what I want to ask of you. All right. So when we look at lymphocytes, there are, as you can kind of see, three major things that happen in the course of their lifetime. They form, right? You're born, you live, and you die, right? So same thing with lymphocytes. Formation of these things. That's the number one event. They have to form. If they don't form, you can't do anything. We don't have an immune system here. We have our primary lymphatic structures, right? And we saw those earlier. Red bone marrow and the thymus, okay? We have our B cells forming there. We have our T cells forming there. Ultimately, we go to the thymus for our T cells to become activated. B cells stay within the bone marrow. They have to be activated. Activation, I mean, it's one thing to form, But it's another thing to be activated and understand what you're going to kill. And the formation of our lymphocytes occurs in our primary lymphoid structures. But until they're out in the real world being exposed to stuff, they don't become active until they get out in the real world. So getting out in the real world is occurring in our secondary lymphatic structures, lymph nodes, okay? Any place where spleen, any place where the lymphocytes are not created, okay? Those are all secondary sites. What happens there? I mean, fluid, lymphatic fluid or blood is going through all these structures, right? Lymph nodes or spleen respectively. That's where they get exposed to the antigen. Once they get exposed to the antigen, then they really become activated, okay? They're going to either, become a T cell or a fully functioning T cell or become a fully functional B cell, all right? And then that's when they will replicate into identical type of other T cells, cytotoxic T cells, helper T cells, B cells, plasma cells, antibodies. And they differentiate and they become identical and proliferate in numbers, okay? That's the second part of their life. Birth, living life, now the response, okay? The response. The effector response is activation to eliminate, okay? Now, they're going to go to wherever they've been exposed to the antigen, either in the lymph node or the spleen. So now they know it's here in their system. Bacteria, viruses, whatever the foreign invader, parasites, whatever the foreign invader is, it is going to release chemicals. And now the body's going to be able to, okay, it's been exposed, so it knows now there's a foreign invader here, and it's going to be attracted to those chemicals. So it'll start to move around. And T lymphocytes, they'll migrate to the site of infection. There'll be a number of monocytes or macrophages or something there engulfing things, trying to kill, can't do it. So those macrophages ultimately will present it to the T cells, and they'll mount the immune response, killing the foreign invader, and then them themselves cease. B lymphocytes, they don't necessarily go to the site of infection, but whatever is in there, the viral infection that's caused their production, or the bacteria, or the allergic reaction, or whatever has caused their proliferation, they stay in those secondary structures, okay? And B lymphocytes will differentiate into plasma cells, which will ultimately result in the production of a large amount of antibodies. B lymphocytes become plasma cells become antibodies. And then antibodies flood the blood, and then they will travel to the site of infection and mount the immune response. And then they're going to do 6 basic things that we'll see, whether it's the variable region or the constant region. There are 6 kind of major actions that antibodies do, and we'll talk about that when we talk about humoral or antibody-driven immunity later on. And this nice little figure from your book kind of shows that off there. Red bone marrow giving rise to all of your B cells or T cells, okay? And you can see they call them pre-T lymphocytes, and I think we'll talk about that here in a second. It's just a non-activated T cell. That's all that is. Naive B lymphocyte hasn't been exposed to the specific thing. It hasn't differentiated into plasma cells. It has not differentiated into an antibody, okay? So that's why they're called pre and naive, all right? But those are the primary structures. B cells, B lymphocytes, again, mature and become, mature in the red bone marrow. Then they go out and then become activated in secondary, but they're created in the bone marrow. T cells, start in the bone marrow, but then they go to the thymus to become mature, okay? Hence B and T, all right? And then you can see the secondary lymphoid structures, tonsils, lymph nodes, spleen, and then what they do, okay? Either helper T cells or cytotoxic or antibodies, plasma cells, okay? Differentiate into antibodies. All the fun stuff that they do. So the last part in dealing with cell-mediated immunity here is just looking at this process, okay? In bone marrow, it all happens there. It's where they're created. They're naive. They go out. They take residence up in lymph nodes and tonsils within the cortex, within the medulla, and they wait to be exposed to some foreign invader. T cells, T lymphocytes, like B cells, B lymphocytes, again, they originate in bone marrow, okay? But they migrate to the thymus, T of thymus, T of lymphocyte, okay? This is where they complete their maturation process. And remember, the great thing about the thymus is it's super active. It's most productive in our youth, and then it just gradually atrophies over the rest of our days. Which is good. I mean, being exposed to a lot of stuff early allows you to create a nice army of T cells to be able to fight things for what you're going to be exposed to later on in your life. But remember, they're kind of that pre-T cell, right, that we talked about in that last little picture there. What does that mean when they're pre-T cells? When they're pre-T cells, they basically have both proteins on it, okay? Both CD4 or CD8. They have them both on there. It's not until, and they all possess a T cell receptor that's kind of randomly, generated on it. And the thymus ultimately decides which cells are going to become CD4 cells and which cells will become CD8 cells. And there's a positive clonal selection. There's a negative clonal selection when it comes to T cells and how the thymus does it. And holy smokes, it's really complex. The thymus really, the bone marrow produces a number of these T lymphocytes, and then they send them out to the thymus to basically come, mature. But the thymus ends up destroying like 98% of them. Most of them don't make it through the maturation process. There's a positive selection and a negative selection that I talked about, and it's through that very complex thing that our T lymphocytes mature, and when they mature, that's when they differentiate, okay? This is the maturing. Okay, and what does that mean? It just means they lose one of their markers on their surface. That's all that means, okay? Helper T lymphocytes keep CD4 protein markers. It's that marker that says you're a helper T cell, and you're going to either help the immune response, help the T cells, or you're going to call in the cavalry and bring more antibodies into the fight because the T cells, the cell-mediated immunity couldn't cut the mustard. Cytotoxic T cells, they keep their CD8. And that's what tells them they're going to become CD8 cells or cytotoxic T cells, sorry. They're going to be filled with enzymatic enzymes, that's kind of a weird way to say that. Various different digestive enzymes, probably the best way to say it. And they're going to be able to create little holes within the plasma membrane of the cell or the bacteria, the virus, or whatever it is. And it will mount an immune response right then and there and kill it. And that's basically what it is, okay? That's the most simplistic way that we can put it. If you're curious, go read the book, okay? But I'm only asking what's on the slides here. So the thymus completes the maturation process. So they're ready to go out. Not quite, okay? Remember, they have to migrate from the thymus to the secondary structures to become activated. we use the word immunocompetent. When they mature, they either become a helper T cell or a cytotoxic T cell. All right, but they're not activated. That happens out in the secondary structures, the tonsils, the lymph nodes, the spleen. They're able to bind to an antigen and respond to it. Okay, that's where they become activated, and that's the word we use, immunocompetent. Okay, bind to the antigen and respond. They're naive. Okay, while they're sitting there within the lymph node, within the tonsils, within the spleen, they are called naive. They're not yet exposed to anything. They don't become immunocompetent until they bind to something. Last thing, there are some regulatory T cells. They call them Tregs. Pardon me for 1 second. All right, I had to sneeze. You didn't want to see that. We have what are called Tregs. And these are just regulatory T lymphocytes. What do regulatory T lymphocytes do? They regulate. Duh. What does that mean, though? It means that they inhibit the immune response. Why would we want to inhibit the immune response? Well, we don't want to bind to self. Okay. It's a form of self-tolerance. It prevents us from attacking ourselves. Okay. It can dampen an immune response to when in a region when we have an immune response occurring. Some tumor cells, some cancer treatments try to inhibit these cells. It's kind of interesting. Some of these, some of these different mechanisms to combat cancer and try to treat it. Some tumors can cause this proliferation of Treg proliferation and cause those cancer cells to not be seen, and the body, we don't mount an immune response against a cell that's not functioning properly. So they're trying, in some cancer treatments, they tried to inhibit Treg activation so cancer cells can't proliferate and we can have a much more quicker targeted response without a proliferation of cancerous tissues. So this is kind of the third T cell, if you will. Helper T cells and cytotoxic, they're the two most important. Regulatory T cells, it's a CD4 cell that binds self-antigens, and it just inhibits the immune response, okay? The stuff that I've got highlighted, that's probably the main things you want to focus on.